Okay.
Hey y'all, my name is Reese Krantz. I'm president of Gulf South Mycological Society, and I'm also on the Korf Foray organizing team. I'd like to thank you all for joining us today for today's presentation on Ascomycetes and the Korf Foray. Before we begin, I'd like to personally thank Dr. Michael Beug for generously donating a signed copy of this book as tonight's door prize, Ascomycete Fungi of North America. Eligible attendees have been entered into a drawing and will hold a raffle at the end of the presentation. With that being said, please join me in welcoming today's presenter, Danny Newman. Danny is an independent parataxonomist and photographer whose work focuses on the systematics of neotropical fungi and diversity of ascomycetes around the world. Over the past 2 decades, Danny's mycological work has taken him to 14 different countries across 4 continents, where he's contributed to the field in many different roles. He's the founder and lead organizer of the Korf Foray, an annual gathering dedicated to advancing the study of Ascomycetes that is now entering its 3rd year. Please join me in welcoming Danny Newman.
Hi, everybody. Thank you, Reese.
Yes, sir.
Can everyone hear me all right? You don't have to see me, but as long as you can see the presentation and hear my voice pretty well, I'm gonna trust. I can't see your responses, but maybe Reese can if people are saying
See
yes.
the thumbs up, thumbs up.
Good, good. So as Reese said, my name is Danny Newman, and I'm the founder and lead organizer of the Richard P. Korf Memorial North American Ascomycete Foray, or the Korf Foray for short, which is much easier to say. Today I'm going to tell you a bit about myself and about the study of Ascomycetes and why they're the best, and about our foray that is now in its 3rd year, as Reese said. So most of the time that I've spent looking for, photographing, collecting, studying, or teaching about fungi has been in rainforests, but specifically Andean and Amazon rainforests. If you've seen me present before, it was probably about Ecuador or Bolivia. One of the reasons I focus on South American rainforests is because of their combination of hyperdiversity and underdocumentation. So as a rule, the more species a place has, the fewer of them we have names for, as in single-digit percentage levels of comprehension. For example, the last time that the total number of described species of fungi from Ecuador was added up, it only went to 3,766 species compared to over 17,000 from the British Isles. And that's not because the British Isles are more diverse than Ecuador by any means. The last time that similar checklisting was done in Bolivia, it was 799 species, which is incredibly, incredibly low. So it seemed obvious to me then, and now, that to go where the biggest knowledge gaps are if you want to have the biggest impact. So while doing fieldwork in Bolivia, my friends and mentees, Elizabeth Melgarejo, now Bolivia's first PhD mycologist, shown here, and Irelis Cuba, shown here, they taught me this word, asco. Pero qué asco, they would say, if I did something gross like eat food off the floor. Qué asco, Danny, qué asco, or qué asqueroso, which means gross or disgusting. And I could never get it out of my head because it had this kind of double entendre for yet another knowledge gap in mycology. Not the gap between tropical and temperate places or rainforests and non-rainforests, but between the two major branches of the fungal family tree, the Basidiomycota and the Ascomycota. So according to one of the more recent great big classification papers cited here, there are now 9 subkingdoms, 19 phyla, 24 subphyla, and 83 classes in kingdom fungi. And together, we have described around 155 to 160,000 species. 2 of those 19 phyla make up 97% of all of those 155 to 160,000 described species. Around 3/4 of them are ascomycetes, compared to around a quarter that are basidiomycetes. And less than 20% of that total are mushrooms in the colloquial sense. And they are from one class, which is the agaricomycetes. So if these proportions hold among the estimated 3 million species of fungi on Earth, then there could be on the order of 2 million undescribed species of ascomycetes. But really, that figure might turn out to be even higher because many of the least studied fungal groups, including tropical fungi, fungal endophytes, microfungi, are dominated by ascomycetes. It will also take us around 5,000 years at the current rates to describe all of that yet unknown fungal diversity. So these are some examples of basidiomycetes. Over on the bottom left, the guild fungi. We've got boletes, which have a spongy porous layer underneath instead of gills. Some of the stinkhorns. Here's are some rusts and some smuts. You might recognize corn smut there. The jellies, the earth stars, and here's a sequestrate or hypogeous basidiomycete in the genus Alpova. These are polypore fungi that look a little bit similar to boletes, but that pore layer is much firmer. The tissues are much denser. They have different kinds of hyphae. You have your stereoid fungi, or as we see a lot of in the tropics, stipitate stereoid fungi that are smooth underneath. Some of the reduced agarics, again, more common in the tropics, but I'm harvested all these from my own photographs, so it's a little biased in that direction. Clubs and corals, toothed fungi, and crusts. And boy, are there many, many, many crusts. And those flashing are the only ones there that are not in the class agaricomycetes. Everything else there that you see is an agaricomycete. And here are some examples of ascomycetes. Again, these might be swayed a little bit more towards the tropics, but the famous cordyceps, sensuato, or the entomopathogenic fungi, which are very charismatic. Ascomycetes make up a ton of plant pathogens. You have the true truffles in the genus Tuber, but then also many other hypogeous ascomycetes. The morels, famous as culinary mushrooms. The discomycetes, so named for having a disc or cup-like shape to them. The other stromatal ascomycetes, like cordyceps, but have these carbonaceous tissues. They're called pyrenomycetes or sorderiomycetes. You might know them as like the genus Xylaria or Daldinia, but there's many, many, many of these. And then various anamorphic fungi, anamorphic ascomycetes, molds. These are fungi that reproduce asexually, or at least they may have multiple stages, and one of them is an anamorph or asexual stage. So for those of us who are more practiced, we might be able to look at a fungus and know, oh, that's an ascomycete, that's a basidiomycete. But really the way to tell them apart is by looking under the microscope at how they form their spores. So basidia form basidiospores, which are typically, but not always, these club-shaped microscopic structures, which are typically, but not always, four-spored, with each spore forming at the end of its own pointy finger-like structure called a sterigma or sterigmata. And then ascomycetes form asci, which are typically, but not always, long, slender sacs or flasks inside of which typically, but not always, form 8 spores. There are many, many exceptions to these generalizations. Asci can be born nakedly, like on the surface of a cup called an apothecium, or enclosed inside of little ascus nurseries that can be flask-shaped, kind of ball or globe-shaped, called parathecia. Both basidiomycetes and ascomycetes can reproduce asexually or clonally. When ascomycetes do it, it's typically called conidia, and there can be different kinds of conidia. When basidiomycetes do it, at least in my experience, I've more seen it called asexual propagules of different kinds. One of them is chlamydospores, etc. Here are some of those typical examples of 8-spored asci with their 8 ascospores, and then interspersed between them are these long, slender, sterile paraphyses. Some of them are smooth and some are kind of finely encrusted, some are more straight, some are more squiggly or curved. So there are plenty of excellent resources that go into as much detail as you could possibly want on the diversity, morphology, biology, evolution, ecology, mating, genetics, and so forth of ascomycetes. And I will end this talk with a very good collection of those resources, but all you need to know for the moment are these very basic basics. So ascos are everywhere. They are in our food, in our farms. In unlucky cases, they're in our bodies or the bodies of other animals or the crops we depend on. But they're also in our medicines and in our hospitals and doctors' offices. They're responsible for everything from acid wash genes to LSD to not dying of the flu. And yet, ascos are frequently found nowhere at your garden variety fungus flora. If they are found anywhere, they are often few in number, relegated to a small corner of a display table, and often given incorrect or outdated names. It's no one's fault, as in it's no one person's fault, not really. These were the most commonly used field guides at the time when I was first learning about fungi, and at best, they dedicate around 10% of their overall length to ascomycetes, and then only the conspicuous ones. Because in order to be paid attention to, apparently you have to be conspicuous. This resulted in a great flattening of diversity, particularly on online biodiversity inventory databases like Mushroom Observer, where every xylaria was polymorpha if it was fat and hypoxylon if it was skinny, and every little cup was Bisporella citrina, and every scutolinia was scutellata, and all Daldinias were concentrica, and every little red dot was Nectrius and Aberina, etc. But if you were a conspicuous fungus, the same would not be true. Meet the charismatic megafungi. The charismatic megafungi are exclusively found in that single class we discussed earlier, the agaricomycetes. The bigger and more edible you are, the more likely you are to get an accurate 3x5 card put beside you at a foray, and the more most people tend to care about you. Fungi and fungus people are not alone in this bias. The trend extends to all life on Earth, not just fungi. The lions and tigers and pandas and puppies and sequoias and sunflowers get the bulk of human attention, while anything deemed too tiny, too ugly, too icky, or too boring gets ignored at best and derided at worst. On the opposite end of this trend is something called Team Tiny, an amorphous assemblage of people anywhere and everywhere who explicitly seek out small, cryptic, difficult, inconspicuous fungi. This is, excuse me, a group photo from the 2011 Spring Fungi of the Sierra Nevada field course. On the left here is Bharati Madhapati. On the right is me, as a much younger man. And Bharati is who I learned the term Team Tiny from and who baptized me into becoming a member during that very 2011 field course. Shortly afterwards, at Bharati's insistence, I raised enough money to purchase the parts that would become the microscope you see there on the right, and I have never been the same ever since. Hiccup. Sorry, here we go. Coming. There we go. One of the reasons to start looking at tiny things is because you've already seen so many of the not tiny ones. The Spring Fungi this year in Nevada course has been held in the same place at the same time of year, almost every year since 1982. The man who founded the course is Dr. Harry Tiers. He's to the left of Dr. Dennis Desjardins there in the photo, and he's standing alone in the photo beside that. And he studied boletes and agarics. His student, Dr. Desjardins, best known for his work on the sometimes tiny, often tiny genus Marasmius, took over for him. And after 4 decades of groups of 20 to 30 people going and collecting fungi from the same habitats, there are still new species being found and added to that list every year. But as time goes on, fewer and fewer of those species will be mushrooms, because the mushrooms are what have already gotten all of the attention. The new professor of the Spring Fungi course is Dr. Brian Perry, who, in addition to being a former student and colleague of Dr. Desjardins, did his doctoral dissertation on the Pyronemataceae, a family of cup fungi in the Ascomycota. That same year, Dr. Perry and his former PhD advisor at Harvard, Dr. Don Pfister, described the discomycete species Chetotirsia vernalis from right near the field campus. Now, this is clever because it's in homage to Hairy Tears. And in Latin, Cheto is hairy, and Tirsia is just his name. So it's Hairy Tears. Very clever. The Desjardins cohort is famous for clever names like these. So Team Tiny is alive and well up in the Sierras. The same idea motivated the late Gary Linkoff to start the New York Mycological Society Christmas forays in Central Park. As one of the oldest and largest mycological societies in the country, NYMS has generated a ton of data about what their mushrooms are during mushroom season, but what they had comparably little data on were the non-mushroom forming fungi in the shoulder or off seasons. And Gary didn't believe in off seasons. So he brought people out to pick up sticks and start looking at the little black dots on them, which were just as much fungus as anything else. Excuse me, but were neglected due to their inconspicuousness and thus difficulty. Gary didn't care. The little black dots went from Central Park to under the microscope and got as good of names as could be applied with the literature available. It should come as no surprise that two of the past three NYMS presidents since Gary's time, Sigrid Jacob and Ethan Krenson, are ascomycete specialists and have each been involved with the Korf Foray in different capacities. And then in 2014, this book came out. It was written by Dr. Mike Beug, who's here with us, with help from Alan and Arlene Bessette. And it was the first major reference on North American ascomycetes in over half a century. And it differed from all of its predecessors by having 843 of its photographs printed in color, captured in situ or in their natural environment. I listened to Dr. Beug give a talk about this book at a meeting of the Pacific Northwest Key Council in 2014, where, if I'm not mistaken, correct me if I'm wrong, he said that in order to be included in the book, the species had to be larger than your pinky nail. It might have been pinky nail or thumbnail, I can't remember. And that still left them with over 600 species to talk about, which is more than had ever been treated in any book of its kind before or since. So now we had an all-color field guide, or at least we used to have one. It went out of print, hence the very, very high prices that everyone was seeing on Amazon. But one book wasn't going to get people to appreciate ascomycetes all by itself. So not long after Ascomycete Fungi of North America came out, I created the Ascomycetes of North America Facebook group as a pretty redundant companion to the outstanding Ascomycetes of the World Facebook group. In 2016, I asked the membership of my group how they would feel about the idea of forming an Ascomycete foray somewhere on the continent. And there was precedent for such forays in Europe, but none in our hemisphere. Folks seemed to like the idea, including several Ascomycete scholars. So 7 years, 2 venue changes, a pandemic, and a catastrophic wildfire later, the Richard P. Korf Memorial North American Ascomycete Foray, or the Korf Foray for short, was born, where all we do is look at ascos, the tinier the better. But who is Richard P. Korf, and why is the Korf Foray named after him? To answer that, we have to go back in time, back through what fellow ascomycetologist Dr. Meredith Blackwell referred to as the Dudley lineage in her genealogy of North American mycologists. Here's Dudley, William Russell Dudley, grew up on a farm in Connecticut, became a Cornell student in 1870, and paid his way through school by milking university cows. He was dorm mates with the future first president of Stanford, David Starr Jordan, in a student-built cottage called the Struggle for Existence, woof. He became an assistant professor of botany and cryptogamic botany, which is kind of what we called mycology a long time ago, before he had a bachelor's degree, and then got his master's in 1876. Was appointed the head of systematic botany at Stanford in 1892. He was a great defender and admirer of redwoods and California flora in general. And he played an important role in establishing national parks in California. He's particularly well known in Santa Cruz, California. He died of tuberculosis in 1911 after contracting multiple illnesses abroad. And Stanford's herbarium was named in his honor when they still had one until it was shuttered and all 850,000 of its specimens were shipped off to Cal Academy in the 70s. And Sarcocypha dudleyi is named in honor of him. This is George Francis Atkinson. Atkinson was born in Rainsville, Michigan. He left home at 13 to become a gold rusher in the Black Hills and a grain handler on the Mississippi River. He enrolled as a junior at Cornell in 1884 and became a student of botany under Dudley. Among other subjects, he graduated with a bachelor's in biology in 1885. And the same year, he was made an assistant professor at UNC Chapel Hill, working mostly on spiders and birds. He was scouted by Cornell bigwigs at the time to replace Dudley as the assistant professor of cryptogamic botany, there's that term again, in 1892, and became department head in 1896. He was a workaholic fungus lover. He only went home to sleep. He rarely took credit on his students' work, and he was perhaps most importantly, a very early and very important and very skilled pioneer of high-quality fungal photography. And I believe, yeah, here's one of his glass negatives, held up there on the left, and then there's a closer image of one of his photographs. I mean, imagine making and taking these kinds of photographs in the late 1800s, early 1900s. And he also promoted the use of one of the earliest color standards. So some of you may have heard of Methuen. Long before Methuen was this color standard I'd actually never heard of until doing research for this talk, called chromotaxia. He was an itinerant neo and epitypifier. So that meant that where a species had been described, when a species had been described, but the type specimen upon which the concept of that species is based, that type specimen goes missing, is damaged, is stolen, and there's no longer a reference to go back to and study and to get a clear understanding of what that species is. You have to go out and create new type collections. And the best, safest way to do that is to go right back to the very spot where the species was first collected, where that type specimen was first collected. And so he did that in Stockholm, England, and France, just to name a few places. And so he was internationally prolific. He died during the Spanish flu epidemic in 1911, and his magnum 10-volume opus was left forever unfinished. The Atkinson collection at the Cornell Plant Pathology Herbarium consists of over 45,000 specimens, including over 1,000 types. Next, we have Herbert Heiss Wetzel, who was born on a farm near Avila, Indiana, graduated from Wabash College in 1902, and enrolled at Cornell immediately afterwards. He worked under Atkinson for 4 years, and then was tapped to become the very first professor of the very first plant pathology department in North America. But he was plucked by New York State College of Agriculture before he could finish his PhD. He maintained a love of and dedication to taxonomy throughout his career, which he didn't have to do, because if you become a plant pathologist, chances are very high, I think in the earliest days of plant pathology as much as now, that taxonomy will not be your focus, but that was something that he was adamant about. He was a pioneer of tropical American mycology, which is close to my heart. And was instrumental in founding the Cornell Plant Pathology Herbarium, to which his 26,000 collections, give or take, were gifted and are maintained to this day. He spent the last 30 years of his life working on a monograph of the Sclerotiniaceae, which he did not live to publish. He was an avid gardener, including plants that he grew specifically for certain rare, unusual diseases that they possessed, that he would show off to people when they came to visit him. Next is Harry Morton Fitzpatrick, who was born in Greenwood, Indiana, corrupted by a young Wetzel who turned him onto fungi as a high schooler. He followed Wetzel to Wabash College in 1905 and then to Cornell in 1908. He got his undergraduate and graduate degrees from Cornell and served as an assistant to Atkinson and eventually became a full professor of the new plant pathology department at Cornell in 1922. He helped found the Mycological Society of America in 1931, was its first treasurer, fifth president, and was its primary historian until his death. He is mostly known for his seminal work on phycomycetes from 1930, and he posthumously published his mentor's monograph of Sclerotiniaceae, Wetzel's monograph, as well as biographies of many important mycologists. He enjoyed playing tennis with his students who knew better than to beat him. And finally, we get to Korf. And I have to start here by saying that there is simply too much to this man's life and legacy to fit into one presentation or publication. So these are merely highlights. For a more in-depth dive into Korf's immense life and work, I have lots of references to share, but chief among them is a talk that was given at our inaugural Korf Foray in 2023, which is available on YouTube, and I'll reference later in the talk. So born in 1925 in Bronxville, New York. It's a typo, I think. Should be Rocks. Oh.
Both mine.
Oh, I think somebody's not muted. There we go. He enrolled at Cornell at 17 years old in 1942, and he intended to become a gentleman farmer. He did not become a gentleman farmer. He studied under both Fitzpatrick and Wetzel, completed his botany bachelor's in 1946 and a PhD in plant pathology and mycology in 1950. He did his doctoral dissertation was a monograph of the Arachnopizzaceae in Lloydia, and he named Arachnopizzaceae Fitzpatricki in honor of his beloved professor. Here's some photos of Arachnopizzaceae species, including the one named after Fitzpatrick. He joined Cornell Plant Pathology faculty and became a Cornell Plant Pathology Herbarium's director, and we think around 1951, became a full professor of mycology by 1961. He was an internationally prolific collector and alpha taxonomist of discomycetes. Discomycetes were Korf's thing. He led field expeditions, which many of his students participated in, to Scotland, Japan, China, Taiwan, Philippines, Caribbean, Belgium, France, Corsica, Hungary, Denmark, Sweden, Norway, Finland, and Micronesia, and then collected extensively in the US, mostly in his home state of New York. He had a vast savant-level knowledge of fungal nomenclature, and for that reason and others chaired the nomenclatural committee for fungi and lichens back when the International Code of Nomenclature for Algae, Fungi, and Plants was the International Code of Botanical Nomenclature. And I want you to pay attention to the family tree here again, because this is how many people Dick Korf trained and graduated. No less than 21 PhD students, several master's students, and postdoctoral fellows, 7 of which would go on to become MSA presidents themselves. Korf co-founded the journal Mycotaxon with his longtime friend, Dr. Gregor Henneberg, in 1974 to expedite and reduce financial barriers to the publication and consumption of systematics research worldwide. He wanted to make it easier to publish, faster to publish, and less expensive to publish. And then just as easy, inexpensive, quick to consume that published information, which was unique and special. He introduced the synoptic key to mycology versus dichotomous key, mostly in the form of these 2 publications, 1972 and 73, respectively. And these standardized definitions of tissue types for a generation of discomycete scholars. He served multiple roles in MSA, including president from 1971 to 1972, and received the Ainsworth Award from the International Mycological Association, the organization's highest honor among many other accolades. He was also an outspoken anti-war activist and protested the Vietnam War specifically He counseled the Cornell chapter of Students for a Democratic Society, and he chaired his local Liberal Party in the 1960s. He was also an avid thespian, and at one point, he was the chair of both the theater arts and plant pathology departments at the same time, from 1985 to 1986. Korf amassed over 5,000 personal collections, which are housed at CUP, including over 250 types. He described and reclassified hundreds of species, published over 400 papers, and he mycologized truly right up until his death at the age of 91. Next, I want to focus on one particular student of Korf's, who is Dr. Teresa Iturriaga. Dr. Iturriaga was born in Venezuela. She got her licenciatura, which is kind of an advanced bachelor's in Latin America, in biology from the University of Caracas. In 1981, she got her master's and PhD from Cornell, both under Korf. She worked on the systematics of the genus Straussmeyeria and Clossinomyces, and the anamorph of Straussmeyeria, Pseudospirophes. And then she stayed on as a postdoctoral fellow. Here are some of the fungi that she worked on. She described Corphiomyces gelatinosum in 2004 from the Venezuelan Amazon in honor of her beloved professor, the doyen of discomycetes, as she called him, and dozens of other fungi from Venezuela and beyond. She was a professor, lab head, curator, and science museum director at Universidad Simón Bolívar from 1989 to 2013 and mentored around 100 students. She was also a member of the International Code of Nomenclature Committee for Fungi. And I met Dr. Iturriaga because if you'll notice, she's wearing a t-shirt there. That is from the, let's see, I think 7th Congreso Latinoamericano de Mycología, 7th Latin American Congress on Mycology in Costa Rica. Well, I attended the 8th one that was in Medellin, Colombia. And while I was there, there were these 2 pre-Congress courses, one on ascomycetes, one on basidiomycetes. And back then I didn't know any better, so I took the basidiomycetes course. But I would sneak into the ASCO one to try and get the best of both worlds. And I worked up a collection of a fungus that, with Dr. Iturriaga, that I still have no idea what it is, would love to know more. And then a few years after that, in 2017, attended the 9th Latin American Congress, where Dr. Iturriaga was also teaching one of the pre-Congress courses. And together we worked up this beautiful little discomycete, which, if I remember correctly, is a velutorhina. And these experiences stuck with me. Both Dr. Iturriaga, both as a person and as a teacher and a scholar, she made a strong impression. So by this time, I knew who Korf was, but for a reason that most people would consider a footnote in the grand scheme of his overall career, which is that he was one of very few people in mycology to rally around the need, behind the need for parataxonomy and parataxonomists. You heard Reese use this term to describe me. This is what I call myself. This is the subject of its own entire presentation, which eagle-eyed viewers will recognize was cited earlier in this talk. But suffice it to say, parataxonomy is a system of labor division applied to documenting of biodiversity, plain and simple. It was first conceived of in the early 90s in Costa Rica by an American entomologist named Daniel Jansen. And like I said, it's what I consider myself, and it's a topic that I have cared about for close to 2 decades. It's formed a significant part of the work of myself and my colleagues in our research on Ecuador, which we published in July of 2023. That is one of 100 different topics I would have loved to have discussed with Korf in person, but I never got the chance because he died in August of 2016 at home, surrounded by family. An event which was followed by an outpouring of memorials and dedications, including an entire issue of Mycotaxon, the journal that was his pride and joy, which you see there on the left, as well as an entire issue of Mycological Progress. And a woman who deserves so much more than a passing mention in this talk is Dr. Lorelei Norvell, who is a towering figure in mycology, who, in addition to writing the lengthy and beautiful tribute to Korf in that special Mycotaxon issue, was the Mycotaxon editor-in-chief for 2 decades, from 2003 to 2023. Here's Lorelei with Gary in the late 90s in Mamma Foray. And that's her in her glass studio as a young woman. And we lost her as well. It was actually just days before the first Korf Foray when we learned of her passing. So in October of 2017, I was living in Syracuse, New York. I was loosely affiliated with SUNY College of Environmental Science and Forestry, and I wanted to get more involved with herbaria fungaria. And at the advice of Lebull-Beniomycete specialist, Dr. Alex Weir at SUNY ESF, I wrote the director of the Cornell Plant Pathology Herbarium, who is herself an esteemed ascomycetologist, Dr. Kathy Hodge, to ask about the possibility of becoming an intern at the herbarium. She told me my timing was good because CUP was about to get a new curator, one Dr. Teresa Iturriaga. Over the next year, Drs. Hodge and Iturriaga and I drew up plans to formalize the creation of a curatorial internship at CUP, with me serving as the program's first intern. My primary project at CUP during my internship was to accession the life's work of a frequent correspondent and one-time co-author of Korf's named Madame Françoise Canduceau. In addition to making thousands of Ascomycete collections, Madame Canduceau was known as being a, quote, difficult personality and, quote, exhausting several husbands. So much so that rumor has it that t-shirts were once made which simply read, "I survived Madame Canduceau." I am so disappointed that I can't share a picture of one of these shirts. I'm also disappointed I don't own one, but not only because I haven't been able to find one, I can't even find a photo of Madame Canduceau herself. Needless to say, I too survived Madame Canduceau, but more in the sense of the magnitude of her collections, which you can see here splayed out at CUP, as well as the inscrutability of her French cursive script, which I got better and better at reading over time. This project ended up being too large for just one person to complete, but some work study undergrads picked up where I left off, and now Madame Canduceau's life's work rests neatly and safely inside the stacks at CUP, available to any and all researchers to borrow in perpetuity. So even though I was too late to meet or apprentice under Professor Korf, I had the next best thing, which was hours and hours and hours spent in the mycological cathedral where he worshipped his beloved discomycetes for almost his entire career. And especially those hours that I got to share with that cathedral's current curator, Dr. Iturriaga. She is who delivered that presentation I said I was going to reference at our inaugural foray, which I highly, highly recommend everyone go and watch. In that talk, Dr. Iturriaga says that you cannot help but absorb Korf by spending time in CUP. And she is absolutely right. The year that I got to spend there afforded me many opportunities for that absorption of Korf and all the mycological predecessors I spoke about that came before him. And this is the primary reason why I decided to dedicate the first, and so far only, Ascomycete foray in the Western Hemisphere to Professor Korf. For another in-depth inside look at the Cornell Plant Pathology Herbarium, there's yet another YouTube video, which I would also highly recommend in that thumbnail there. At last, that brings us to August 4th, 2023, where after several years of planning, several difficult personalities, including my own, several last-minute changes, one legal demand letter, one global pandemic, and one catastrophic wildfire, the first Richard P. Korf Memorial North American Ascomycete foray was born. The first Korf Foray was a 2-day, 3-night Ascomycete-focused taxonomic retreat located in the heart of the hyper-biodiverse temperate rainforest of the Blue Ridge Mountains in Highlands, North Carolina. Those twin knowledge gaps, mycological knowledge gaps that I mentioned close to the start of the presentation, ascomycetes and tiny things and rainforests, the goal was to combine them into a single event. The foray is modeled after the meetings of the Pacific Northwest Key Council and the Ascomycete field workshops of Northern Europe. More on those in a bit. And it's the first event of its kind that we know of in the Western Hemisphere, a place where teeny tiny are the rule, not the exception. Another thing that makes the Korf Foray unique is that participants are expected to possess an intermediate to advanced level of skill, both in the field and the lab, and the equipment and techniques associated with same. So there are beginner and entry-level events all across the United States and beyond, but there's really very, very few places for that intermediate and advanced level. We are specifically trying to fill that niche. We also follow herbarium quality collection documentation protocols because all of our specimens are getting deposited into Fungaria. And these will be familiar to anybody who's worked with fungi for long enough. So we had 4 kind of core components to the foray. The first one being the field. The goals here were to have immersive, long-form, multi-site collecting trips to a curated selection of pristine, accessible, fungus-filled habitats. I think our participants would say we achieved that. Next is the lab component. Open-ended lab sessions with well-equipped workstations suitable for collaborative or independent microscopy. We got to use this really wicked dissecting scope connected to a big screen TV where we could kind of show off things to folks that we had found. Collaborative or independent microscopy, photography, computing, culturing, sequencing, or the task of participants choosing. Next was leisure. We, I think, hit a home run here as well. The Highlands Biological Station was a very, very pristine, special, relaxing place to be, even when not mycologizing. Picturesque grounds, campfires, lakeside strolls, sunsets, starry nights. A little mushy here, but ample refreshments, custom catering, abundant local and regional recreation. And then finally, we had a lecture series as well. We had 4 invited mycologists, 2 of which sadly could only attend virtually, gave their lectures over Zoom, 2 attended in person. Dr. Ituriaga was the first of the 2 in-person invited mycologists, and she gave her talk on remembering Professor Korf. We also had Dr. João Araujo, who at the time was the assistant curator of mycology at the New York Botanical Garden, who is now at the University of Copenhagen in Denmark. He's well known for his work on entomopathogens or cordyceps in the loose sense. He gave us a talk on the biology behind the zombie ant fungi. And anyone who has seen Joao's presentations know that they are some of the best. They put mind to shame. And then attending remotely, we had Dr. Luis Quijada, who was a postdoctoral researcher at the time at the Pfister Lab in Harvard University, excuse me. And his talk was entitled Past and Present of Inoperculate Discomycete Studies in the Canary Islands, where he is from. And lastly, we had Bjorn Vergen, who gave us a talk on rare pyrenomycetes. And we very much hope to see both Luis and Bjorn attend a future Korf Foray. Altogether, we had 22 participants representing 3 continents and 4 countries, the US, Germany, Brazil, and Panama, including Mark Stadler, shown here, who is the head of the Stadler Lab at the Helmholtz Center for Infection Research in Braunschweig, Germany, and is also the current president of the International Mycological Association. We had Arkansas Mycological Society co-founder and author Jay Justice, who pretended to like Ascos for a few days with us. Duke lichenologist and former CUP curator, Dr. Scott LaGreca, my good friend from our work together in Ecuador, Dr. Ru van de Grift, visiting us from his job with the USDA Animal and Plant Health Inspection Service, and those two New York Mycological Society presidents I mentioned before, Ethan Crenson and Sigrid Jacob. 3 of these attendees applied for and were awarded the Kumi Korf Scholarship, named in honor of Professor Korf's wife of 50 years, whose generous contribution to the first Korf Foray we literally could not have done without. The recipients included Connor Dooley of Oregon State University, Judson Van Wyck of the Bonito Lab at Michigan State University, and Marjorie Cedeño of the Stadler Lab, previously mentioned. Multiple other attendees donated to that scholarship fund as well, and we are very grateful that some, like Cornell faculty Mary McKellar, shown here, continues to do so each year, whether or not they even attend the foray, which is very sweet. As a bonus for anybody who registered for the main Korf Foray, we threw in free attendance at what we were then calling the Purchase Knob Ascoblitz, located at the Appalachian Highlands Science Learning Center inside of the Great Smoky Mountains National Park. So the first section, the sort of Korf Foray proper, the first year that we did it, was not inside of a national park, except for this kind of add-on, which was inside of a national park. And at this really, really stunning field station. A huge part of the reason that we were able to offer this sweet add-on was due to a contribution from Discover Life in America, who, among other things, are the folks behind the Smokies All Taxa Biodiversity Inventory, or ATBI for short. In total, that year, we generated 270 records, which correspond to 228 voucher collections, representing 92 genera, 66 families, 31 orders, 12 classes. And our master database and an accompanying abstract were published in the very last issue of Mycotaxon before, sadly, the journal closed its doors for good, being unable to fill several critical roles following Lorelei's passing. The good news is that the journal has since been 100% digitized and is made entirely open access. And the Korf Foray Regional Checklist, which we published, has a permanent home on the mycobiota section of that Mycotaxon website. We also created a continuously updated corresponding IMAP project. And the only other Korf Foray related peer-reviewed publication we're aware of at the moment is this one from the Stadler Lab, which epitypified Hypoxylon perforatum with material that was collected during the Korf Foray. And there you can see us getting a nod from them in the acknowledgements. We did it again in 2025. We needed a year break after the first one, but this time much simpler to the improved sanity of all involved. Some of our special attendees were, sorry, let's see here. There we go. Flavor and aroma chemist at the University of Tennessee, Knoxville, Dr. John Manafo, along with 2 of his students. We had Patrick Verdier, a former student of Korf's, who found out about the foray through some sort of backside channels and wrote us out of the blue. And he brought with him one of the most incredible microscope setups that any of us had ever seen, which we will have shots from shortly in a moment. We also had entomopathogen or cordyceps specialist, Dr. Richard Tehan from the University of Utica in New York, and Maricel Patino, who is Portugal's Michael Jordan of amateur mycology and a dear personal friend. We awarded 2 scholarships this year, one to Kurt Miller, who's an independent mycologist like myself from Puerto Rico, and to Warren Cardamona, who's a master's student from the Perry Lab at California State University, East Bay. And this time we got a picture of them getting their scholarships. This field station, the Appalachian Highlands Science Learning Center, is now the sole location of the Korf Foray because we like it too much. It's pretty perfect as far as field stations go, and I've been to a handful of them. So this 2025 foray, as well as this upcoming 2026 one, it's designed more in the style of a biodiversity expedition, as opposed to something more meticulously programmed or scheduled. And this is the format we're sticking with for now. We hope to bring back a lecture series in future forays, but for now they are on hiatus. We added in 2025 another 203 records, 192 of them Ascos. Yes, we do accept some Basidios, corresponding to 198 vouchered specimens, several of which have been determined to be new or very likely new to science. I am now finally going to talk about some of those interesting, possibly to definitely new fungi that we found from both years, from 2023 and from 2025. This is a fungus that we found only after recognizing it from Bjorn's excellent rare pyrenomycetes talk, which he called his, quote, all-time favorite pyrenomycete, which coming from Bjorn really means something. He was referring specifically to Wintiomyces fimbriatus, and noting that it is very, very rare, which also means a lot coming from Bjorn. It is only 80 microns in diameter. It is a tiny, tiny, tiny fungus. I've tried to give you a visual aid there to see what it is we're looking at. We saw his images in this presentation and we looked at one another and said, I think we found this. It was essentially bycatch. The collection, the target species for this collection was the little red balls, the nectriaceous looking thing. And then we just sort of accidentally noticed, hey, wait, there's that thing that Bjorn said we should keep an eye out for. This is Flammocladiella anamiae, which is in a genus of just 2 or 3 species. And it's the only genus in its entire family, Flammocladiolaceae. This is friendly fire between ascomycetes. The host is this, so you have the fuzzy white-orange balls on top, and then this larger paratheciate fungus underneath. The fungus that's being parasitized is a species of Messeria, which forms its own black and white parathecia inside the wood, and then sends a bunch of little black chimney-like structures up through the bark called osteolar necks. And then the Flammocladiella is the orange, white, fuzzy balls that are formed on top of those structures and parasitize it. This genus was only described in 2015, and the species was only described in 2019 from Europe, and it's known from just a handful of records worldwide. These are some of Patrick's excellent micrographs, at least one of which is utilizing DIC or Nomarski, which makes for some really three-dimensional looking micrographs. We'll be seeing a lot more of those micrographs in the coming slides. This is the new species of Genia, and it's a hypogeous or semi-hypogeous truffle kind of thing. It's not a true truffle. It's not a culinary truffle, but it's an ascomycete ball type of thing that grows underground or undergroundish. It's got these very, very, very ornamented spores. And we have it on good authority from the specialists in the group that this one is a new species. This is a Pesicula species, which we found a few examples of at Korf Forays, but none with this weird anamorph attached to it. So Patrick has done us the benefit of kind of pointing to what is the Pesicula cup fungus, the teleomorph, the sexual state, with green arrows. And then the black arrow, those like long chimney flask type of things, that is apparently an anamorph, and has stumped just about everyone. What you're seeing in some more excellent micrographs from Patrick are the staining reactions of the tip of the ascus in the presence of different chemicals or combinations of chemicals. That's that micrograph of his that's got the 2 asci on bottom with the blue tips, and then above that, the red tips. For a great, great many discomycetes, we look at the material first in tap water to see how the structures look while they are still alive. And this is called vital taxonomy. And it could very easily be the subject of its own presentation. I'll have a link to Hans Otto Boral's webpage on vital taxonomy at the end of this talk. Zotto, as he is colloquially known, is the reason that we know that this matters. And Korf implored people to take his vital taxonomy seriously at a time when it was still new and highly controversial. This is Pithiella hispida. Pithiella is a genus that was redefined by Korf and Zhuang in 1987. And this species specifically was described from Baja California in 1994. It seems to like acorn shells, which is what we found it on. We're grateful to Czech mycologist and symphony musician Victoria Halasu, I'm probably not pronouncing that right, and upcoming Korf Foray attendee Francois Guay, which I may also not be pronouncing correctly, for identifying this for us. Because there are no reference sequences of this species in GenBank, sequence validators on INAT arbitrarily gave this a provisional name in the genus Eriocephala, but not taking into account the very distinct micromorphology, in particular, this unusual combination of slender asci and almost completely circular, like globospores. The only records of the species that I could find outside of our collection and the Mexican type are one of Francois' in Quebec, and maybe one or two others. This is a new species of Xylaria, which was found at both the first and second Korf Forays. The first time that we found it on the base of a beech tree, and it was impossible to find spores. We tried and failed under the microscope, could not find spores, mature or otherwise. And I had thought that the purple might be a virus or a secondary fungus because I've seen some Xylarias that have this look to them, and that's the reason why. So I sent it off to Rue, who some of you may know is a specialist in Xylaria. He also couldn't find any spores, and he didn't see signs of an infection, but he couldn't really say for sure. But he thought the purple pigment was natural. In 2025, Fenner Martin Morse attended our foray. We showed him these pictures, and right away he says, "I bet you found these around the base of a beech tree and had a bear of a time finding spores." I'm like, what? Yes, exactly. He said he knows the species. It always occurs this way. The purple coloration is indigenous to the fungus, and he has provisionally termed it Xylaria circumdendrophila, for it likes to encircle trees. This is Dasycyphella chrysotexta, originally known as Velibidia chrysotexta, a genus that was only described in 2019 and named after the mountain it was found on in Croatia. There is some disagreement as to whether it deserves its own genus or not, but what is for sure is that there appear to be very few records of it from anywhere. And now we have one from the Smokies. And you can see in one of Patrick's monographs, and also in Kurt's lovely drawing, that the tip of the ascus is amyloid in the presence of iodine. This is Rodwaya sessilis, which we have seen on rhododendron on two separate occasions. Virtually every habitat we're in, up in the Smokies, Blue Ridge, there's rhododendron somewhere. Some forests are dominated by it. This is one of only 4 species in the genus Rodwayella, which was erected in 1986. Rodwayella sessilis was first described as a holotium in 1925, then moved to Pisicula, which is what we thought this was initially, in 1958, and then Rodwayella in 1987. And it's not 100% clear if our species is the same as what's found in New Zealand, but regardless, this is either rare or rarely reported for sure. Here's another example of vital taxonomy in action. The above 2 micrographs are in tap water, and the bottom 2 are in Lugol's, which is an iodine-containing reagent used for discomycetes. And notice, if you can, how the gutules, the little oil droplets inside the spores, are present in the living tissue and then are absent in the dead tissue. More friendly Ascomycete fire. I mentioned cordyceps at the beginning of the talk. There are lots and lots and lots of cordyceps or entomopathogenic fungi in rainforests, and the Blue Ridge and Smokies are no exception. In this instance, we have a parasite of a parasite, also known as a hyperparasite, And this one is in the genus Cordyceps. And if I'm not mistaken, we think that this one is also new. The fungus that eats the fly or the ant, the cordyceps or Ophiocordyceps or metacordyceps or what have you, this is the fungus that eats that. And it was Dr. Richard Tehan of Utica University who collected 2 out of 3 of these. Connor Dooley collected the other one, and he'll be the one to let us know if it is in fact new to science. This is another enteropathogen we found in 2023 in the genus Gibellula, which is exclusive or almost exclusive to spiders. And I have cheekily given it an informal name, Gibellula libum natali, in the event that it turns out to be new. Libum is Latin for cake, I think, and natali of birth, so birthday cake, Gibellula. This is Pseudosclerococcum golindoi. In addition to being a mouthful, it was described in 2019, and until this collection was made, it was known only from the type collections made in Basque Country in northern Spain. It is incerticetus, which means that it is of uncertain placement in terms of what family it belongs to, but it's in the order Sclerococcales, which, suffice it to say, is very distantly related to just about every fungus we've discussed here so far. It's strange, it's beautiful, and this may be the first time that it's been found in the entire Western Hemisphere. This is Ionomydotis olivacans, which is part of a family, a favorite family of mine, the Cordiaritidaceae. To be ionomydotic is to have pigments in your tissues which are extractable with potassium hydroxide, or KOH. And the way to check for this before going to the microscope is to place a bit of tissue inside of a porcelain spot plate. You can really use any sort of non-porous white surface like a plate. And then put a drop of KOH on it and see what colors emerge, when they emerge, and what they change into, if they change versus staying the same. I wish I could show you some spot testing for this collection, but we don't appear to have photos of that. But this species was described by Durand in 1923 from Coconut Grove, Florida. And indeed, what records there are for this species, they are all subtropical to tropical, except for ours. According to Zato, there might also be one from Japan, which would make sense because there is a strong biogeographic signal between the rhododendron mountain forests of the US and of Asia. And another example of that is this truffle. It's a hypogeous ascomycete that's not a true truffle like the culinary kind, but still a truffle nonetheless. And despite what it looks like, it's a relative of the morels. This is the genus Imaya. It was erected in 2008 by Trappi and Kovacs, if I'm saying that name right, in honor of Japanese mycologist Dr. Sanshi Imai. When Korf first traveled to Yokohama, Japan in the 1950s, he was there to meet and work with Dr. Imai. And it was where he met and fell in love with the woman who would become his future wife, Kumi Korf. It is therefore very auspicious then for us to have found this genus, which is only known from Japan and the southern Appalachian rainforest, another expression of that strong biogeographic signal between the two biomes. Imaya pachyascus and the very recently described Imaya kubohiensis are the names given to the North American species, while the Japanese one is Imaya gigantea. This is, I'm not going to tell you what this is, not yet. It was picked up by Kurt Miller, one of our scholarship recipients from 2025, thinking it was some kind of truffle, some hypogeous thing, whether the Asco kind or the Basidio kind, which can be very difficult to tell apart which is which without a microscope. And this was a case where sequencing really came to the rescue. We were stumped without it. When we sequenced it, turns out this is Pseuilus spraguei. This is that mushroom. These are the same thing. Pseuilus spraguei sometimes apparently likes to form these large nodules on the roots and rootlets of the conifers it mycorrhizally associates with. I had never seen anything like this before, and I don't think anyone else had it before. So it definitely, it passed muster, it became a collection. And finally, I have to end on this quadripartite interaction between no less than 4 different species of fungi, 3 ascomycetes and 1 syphaloid basidiomycete, which are some of the only basidios that we welcome at the Korf Foray on account of how hard they are trying to impersonate ascomycetes. So first you have the Merismodes, which is we think is Merismodes confusa, which is the syphaloid or syphaloid basidiomycete. And you've got this, I have these out of order in my notes, forgive me. I'm gonna just read what I have. So you have this Diatripella species, that's what the taller arrow is pointing to. And that's a fungus that, like the Masseria I mentioned in the Flammocladiella slide, forms these black and white parathecia inside of the wood, in this case birch. And then the syphaloid, which is on the left, shorter arrow, grows on top of that fungus. Is it parasitizing it? No idea, maybe. Intermixed with the Merismodes and is a Dialonectria species, the little red nectarioid balls. And on top of those balls is growing a Hyalobilia erythrostigma in another possibly parasitic, possibly incidental relationship. That is a fungus on a fungus on a fungus on a fungus. If everything going on here is parasitic, the Hyalobilia is a hyper parasite. Top that, I challenge you. So what does the future look like of the Korf Foray? We have our 2026 Korf Foray, which is real close to starting. It is July 27th to August 4th of this year. Once again at the Appalachian Highlands Science Learning Center in Purchase Knob, North Carolina. We're very lucky this time to have with us Chef Sean Thompson. I cannot tell you how valuable it is from experience to have food taken care of by others while doing this kind of work. It frees up so much time, and we look forward to having Sean with us, not just as a great chef, but also as a participant. He's interested in taking part in as much of the foray as he can. For next year, we're not sure when the foray is going to be, but it's not going to be during NEMF. We have heard your pleas, and we will not have schedule conflicts with NEMF. We might have schedule conflicts with something else, but not NEMF, but we plan to host it at the same location. There are these 2 grants that we've been encouraged to apply for by our friends at Discover Life in America, one for $7,500, one for $15,000. And we will be writing applications for both of those. I also in the past have had good success with experiment.com, which is kind of like the Kickstarter or a science version of Kickstarter. So we have an experiment.com campaign that is in the works. And then what do we do when we get all that sweet, sweet money? Well, we have more forays. We'd like to invest in some kind of permanent foray microscopes, a glamping tent to put on our tent platforms at the Appalachian Highlands Science Learning Center. Maybe not spend money, maybe save some and keep it around to spend when we need it. It'd be sweet for me to pay my co-organizers. I would consider myself the last person who should be paid. As of right now, this is an entirely volunteer run, no money, no salary kind of job, other than getting your attendance comped. We could do with a revamping of the website, although we've gotten a ton of really valuable help from Ethan Princeton on web development up to this point. We want to offer more scholarships. We would be cool to hire a grant writer, but we gotta get a grant to hire a grant writer, so a little bit complicated there. And then who knows what else? But we want to definitely keep this going. So you remember those European Ascomycete forays I mentioned? I went to one in 2024, and it turns out that everything that we do, they have been doing it better and for longer, including the wine and the Genepi. These are photos from the 2024 ascomycete.org Alpine Zone Foray held in the French Alps near Vallorcine or Chamonix. And I am deeply indebted to Nicholas Van Voren, who was pictured in those last images, in particular for ensuring that this helpless Yankee and his 150 pounds of luggage could successfully attend that foray. Nicholas and his fellow Ascomycete.org co-founder Michel Hérault, I'm saying that wrong, I'm positive, are also responsible for donating the 10 or so copies of the superb reference studying the discomycetes, which we have on hand now at every foray. Another main reason I'm bringing this up is because the folks up at ascomycete.org put together a very handsome report summarizing their findings from each of their alpine forays. And thankfully, Nicholas et al. Have agreed to help us produce a similar report that will cover all of the cumulative data from our 3 core forays to date sometime later this year. And here's a group photo from that foray in 2024 up in the mountains. Oops, it looks like that happened twice. There we go. Lastly, the future is you at a future Korf Foray. Please take an interest in these hyper-diverse and understudied, underdocumented creatures that we share the spinning ball of juice in the sky with. Buy a microscope and a camera with a macro lens. They are cheaper than you think. Borrow or buy and read the resources I'm about to share with you, and join and participate in the groups that I'm also about to share with you, and then come Korf Foray with us. You will not regret it. With that, I would like to acknowledge all of the sponsors that I also had listed at the beginning of the talk, as well as these special thanks, and especially to my Patreon supporters named there. I've given a link and a QR code if you would like to become one such supporter. All the images used in this talk belong to me and have a Creative Commons, non-commercial, no derivatives attribution license, which basically just means you can use them for free if you're not making money off them, and it's for, educational purposes, except where I've indicated in captions that the pictures belong to somebody else. And I thank those people for allowing me to use their images. And last, but very, 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 very far from least, I have to thank my 2 very special co-organizers, Reese Krantz and Emma Donkowski, without whom no Korf Foray would be possible. So thank you so much from the bottom of my heart. Here are the books I'm going to recommend. These are not all the ASCO books that one could buy, not by a long shot, but I think if you had all these, this is a pretty darn good start. This talk is being recorded. If you can't screenshot this or write them down, don't worry, you'll have other opportunities. Same with this. These are a list of web resources and a list of specialists, mostly kind of folks I could rattle off the top of my head. I don't have a works cited page for you. It was a miracle that I got this done when I did. Please forgive me. I will give this to anybody who wants it later. And finally, we have come to the reason you're all really here, which is who's going to win this dang book. So, Reese, are you doing the drawing?
I'm here. Actually, I think it'd be fitting for Emma to do it. Emma, are you here?
Yes, I'm here.
Okay.
Wait, let me maximize this. Open this up. Can you hear me?
Yeah.
Mm-hmm.
Can you see me though?
Yeah.
Yeah.
Okay, I have my witch hat and I have everyone's name in the witch hat. And we will find out who won. I feel like I need like a drum roll or something. Mike Garton is the winner.
Mike Garton.
Hey,
Dun, 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 dun.
congrats, Mike.
Woo!
He's actually he's in the group too.
Yay.
No.
Holy crap.
Holy crap, you won! Whoa.
Oh, thank you so much. This is awesome.
Yeah.
Wait,
Congratulations, Mike.
Oh, thank you so much. This
We
is
hope
incredible.
you will use it and join us with your book at a future Korf Foray.
We'll see. Well, maybe.
All right. The book.
So, does anybody have any questions before we close out? Any ASCO curious people?
I realize that I opened up a fire hose of info, so it's a lot to take in. But well, I think folks know where to find me if they have any questions they want to ask at a later date.
We want to see the resource slide real quick before we
Resource
close.
slide, sure.
Yep.
The book one or the one after that?
I think it's the one after that.
This one. Okay.
Well, and Danny, I just want to say it's good to see your face again. It's been 14 years.
Something like that.
Well,
Yeah.
no, I'm 12 years. I'm sorry.
12 years. Yeah, long time. I'm real glad you could make it, and I'm also glad that you donated this book and made this whole talk possible. So thank you.
Well, it was an accident, and Korf was very important contributor helping me figure out how to write this bleeping book.
Yeah, I'd love to hear more about that. Yeah, if you don't mind sharing, what was his participation like in the creation of that book?
Well, he contributed to many different chapters, and he told me the story about how then Gyrometer Korfii got named to scold him, not rather than to honor him, because he thought it was at the time the same as what was then Gyrometer Gygus. So, but he was just a great person to work with. But every single specialist Ascomycete in Ascomycetes reviewed their specific chapter. And so I called this final book my symphony. I was just the conductor. I wrote it because I didn't know anything about Ascos to start with.
Right. Well, you could have fooled us because it holds up. I've got a copy. I suppose if my copy ends up being worth like $10,000, I might have to consider selling it, but otherwise I'm hanging on to it for indefinitely. Any questions that popped up in the chat or
I don't
not?
see any.
Okay.
Go ahead and close. Thank you, Danny.
Well, thanks everybody. And yeah, come Korf Foray with us.